comparative neuroanatomy. Hello, my name is Katja Heuer, and I will be talking about phylogenetic comparative neuroanatomy. And the data and resources to get started with non-human data will be covered by Austin. The main objective of my talk is to highlight the importance of considering phylogenetic relationships when comparing brain traits across different species. And by understanding these relationships, we can interpret neuroanatomical data in a more accurate evolutionary context and gain clearer insights into brain evolution. The study of brain evolution provides us with a unique perspective to understand normal and pathological variability in humans. Across mammals, the human brain is extremely variable in size and shape. And if we look beyond human brains, across non-human primates, or even across mammals, there is an incredible neuroanatomical diversity. Interesting to put the human brain in its evolutionary context. And if we look across vertebrates, it gets even wilder. There is a sneak peek into our growing brain catalog collection. And comparative neuroanatomy is mesmerizing at many levels. So from the macro scale in brain shape to the micro scale as shown in these Brownian maps, where cortical areas are shown with different symbols. And these are based on microscopic images looking at cell density, cell shape, the layers in which cells are organized, and the thickness of these layers, and the presence or absence of them. Now, what drives the interplay of variability and stability, of diversification and conservation? So in comparative neuroanatomy analyses, we aim at comparing brain structures across various species to understand functional and evolutionary differences. However, treating each species as an independent data point can lead to incorrect conclusions because they're not independent. And they are all linked by the tree of life. And these foundations of comparative studies are deeply rooted in Charles Darwin's theory of evolution by natural selection, where he emphasized that species groups into genera evolved from common ancestors and are related to each other and also extinct forms in that tree of life. And that highlights the importance of evolutionary relationships. And this branching phylogenetic tree represents our understanding of how life evolved from one common ancestor to all living things as an array of splitting events. And the shape allows us to see how species are related to each other and which branches took off into an enormous diversification or which groups remained rather stable. And everything living is related. And so we use phylogenetic comparative methods which help us account for this shared evolutionary history. And this is super important because species traits could appear as correlated just because they split from a recent common ancestor. Take this scenario, for example. It's adapted from Felsenstein, which is a really great book if you want to learn more about phylogenies. So from a common ancestor with a certain trait, there's a split into two species. So the ancestral trait is varying randomly according to a Brownian motion model. And until this point, it keeps on varying randomly. And then there's the split, and it keeps on varying independently in both lineages. And so shortly after the split, it's still quite similar. But then after more and more time has emerged, the traits can become very different between both species. So one is down in the morphospace here and one is up here. And then at this point in time, so not very long ago, there's a new speciation event into several new species in each branch. And so from there, the trait varies randomly here, as you can see, for each of these starting points. And so now you measure your phenotypes across these species. And if you do not take their shared evolutionary history into account, you may have the impression that there's a strong correlation. However, if you account for phylogeny and you do a phylogenetic regression rather than a positive correlation, you will observe that there's actually no correlation. Now, how do we do this? So one element we need for this type of analyses is the phylogenetic tree of the species that we'd like to compare. And this is just an example from a recent study we did across 56 different mammalian species. Such phylogenetic trees can be built based on differences of homologous genes and or traits across species, like morphological traits or other types of data. And the shared evolved characters, meaning like new traits that are common to all species in one clade, but not present in those close relatives of another clade, they allow us to see which species were formed from a common ancestor. And then the amount of edits needed to transform one gene sequence into that of another species enables us to estimate the timing of when they split from their common ancestor into the new species. And so the resulting trees can then not only represent the relationship between species, but also along all ancestral nodes of the tree, but also encode the timing of the splits into their branch lengths. And so you can obtain phylogenetic trees for exactly the species in your samples from several online sources, like, for example, the time tree of life, which is also gives you some additional information on events in geological time, astronomical history, oxygen levels, and other events, and the 10K Trees website, which is specialized on primate phylogenies, among others, and so they have the most primate species included to date. And phylogenetic comparative methods use such phylogenies to build and test different models of phenotypic trait evolution. And the three most common models are the Brownian motion model, the Ornstein-Uhlenbeck model, and the early burst model. So the Brownian motion model is one of the simplest models of evolutionary change. It assumes that random diffusion of phenotypes changes from tips along the branches of the tree, with changes occurring in small random increments over time, so that the earlier two species split from one another, the more they are supposed to be different. And so the Brownian motion model can be used to model fluctuating selection and genetic drift, for example. And once an ancestral species splits into two new species, their phenotypes will become progressively decorrelated. And if you'd like to run some simulations mathematically, this model can be represented like this, where you have the infinitesimal change in the trait X at time t. And this here is the magnitude of the change in the trait at the time, and so also known as the diffusion coefficient or the evolutionary rate parameter. And so a larger evolutionary rate parameter implies a more substantial change per unit of time, which then will lead to a greater trait variability. And then you have the scatter generator, so for making the random fluctuations. And the Brownian motion model assumes that there is a mean of 0 and the variance of 1. And over time, it ensures that the trait changes are randomly and normally distributed. And so the model could model genetic drift and represent neutral evolution, but it could also be due to adaptation if it happens very fast. The Brownian motion model assumes that trait changes are independent and normally distributed, and that the variance of the trait changes is constant over time. And here we can see the effect of time on the phenotypic evolution. So with the same starting value of 0 and the same evolutionary rate parameter, trait values of species diverge more and more and more over time due to the random fluctuations. And so the variance among runs of this process increase linearly with time. And here we can see the effect of the rate parameter. So a higher rate parameter means an increased magnitude of the trait changes per unit time, and that creates a larger spread of trait values over the same amount of time. And to properly account for phylogenetic relationships in our analyses, it does involve converting the phylogenetic tree into a variance-covariance matrix. So a phylogenetic covariance matrix describes the expected variance and covariance of traits among the species in our sample based on their shared evolutionary history. Each row corresponds to one of the species in the tree, and the entries in this matrix are calculated based on the branch lengths of the phylogenetic tree. So each tip value can be thought of as an ancestral value plus the sum of two evolutionary changes. One is the shared branch length between two species, and the other one is the unique branch length that's only for each tip species unique. And then you compose these for the entire tree, and in your matrix, the diagonal elements represent the evolutionary path length from the root to each species, and in the off-diagonal elements, you have the shared evolutionary path length between the pairs of species. And next, we need to estimate the phylogenetic signal in our data, the so-called Pagel's lambda. it indicates how much the phylogeny predicts the covariance among the trait values. So the extent to which related species resemble each other more than they resemble species drawn at random from the tree. And a lambda value of 0 implies that there is no phylogenetic signal. Traits evolve independently, so it's like basically a phylogenetic regression would then mimic just a normal regression. And a lambda value of 1 implies that trait evolution is fully explained by the phylogeny as expected under the Brownian motion model. And then you use this in your regression, so the lambda multiplies the off-diagonal values with its value, so between 0 and 1. And if it's 0, they turn into 0, and you can your phylogenetic least squares would be just a normal regression. If we want to consider more than one character at once, we need to use multivariate models. So in addition to the phylogenetic variance-covariance matrix, we need a matrix for the variance and covariance across traits due to their tendency to evolve together. And the diagonal entries in this matrix provide estimates of the net rate of evolution for each trait, and the off-diagonal events present the evolutionary covariance between pairs of traits. And then to get the variance-covariance matrix, that describes all characters across all species, we combine these two matrices into one single large matrix using the Kronecker product. So where you multiply the matrix by each of the elements, so that a 2x2 matrix would turn into a 4x4 matrix. And if you'd like to learn more in detail, I highly recommend this wonderful open access book by Luke Harmon, referenced here. So another major model is the Ornstein-Uhlenbeck model. Under the Ornstein-Uhlenbeck model, phenotypes are still assumed to vary randomly, but the model supposes an adaptive threshold towards which these random phenotypes changes are attracted and stabilized towards, which is in our example somewhere above here, up here. And these could be particularly favorable values, which have a higher chance of being selected for, and the strength of the selection determines how strongly species are pulled towards the optimum. And here in our example, this optimal value is here. And the Ornstein-Uhlenbeck model extends the Brownian motion model by including the idea of an optimal trait value. And so if you like to run some simulations mathematically, this model can be represented by this equation where this theta is the optimum trait value. So depending on the particular context, it's either positive or negative to attract the phenotype. And the alpha is the strength of the attraction to the optimum. And the higher alpha indicates a stronger pull towards the optimum. And then traits will vary randomly with the random component as in the Brownian motion model, but will also be attracted to the optimum value, which has been used to model stabilizing selection processes. And finally, the early burst model. It assumes an initially fast rate of phenotypic changes during the time that a new adaptive regime is first invested, early in a clade's history, which then later slows down. And it's also called the accelerating decelerating model, and it's an alternative extension to the Brownian motion model, where the magnitude of random change is modulated by an exponential function. So if you'd like to run simulations, this is the equation that would match this early burst model, where the magnitude of random trait changes is modulated by R, so the strength of the exponential change. And so the rate at which the magnitude of changes accelerates or decelerates over time. And when R is 0, it's just like normal Brownian motion. And when R is larger than 0, the variance of the phenotype increases exponentially. So a rapid early diversification. And so it assumes an initially fast rate of phenotypic changes, which later slows down. And it's used to model rapid diversification of species in the early stages of their evolution, followed by a slowdown in the rate of diversification. And so it's often used to explain adaptive radiations. And phylogenetic models use these trees to test the fit of different evolutionary models to your data. And it is always important to test the model fit on your specific data set. In the 3 studies we did, each of the models provided the best fit once. So studying brain size evolution across primates, we found that the changes fit the pattern of a neutral evolution, so suggesting that random changes played a significant role in primate brain evolution. But then zooming into the New World monkey branch, the early burst model provided the best fit, indicating a rapid diversification of brain shapes during the initial stages of the New World monkey brain evolution. And then zooming out into mammalian brain evolution in our latest project, a stabilizing selection process was best describing our data. So likely indicating adaptations to specific ecological niches with strong stabilizing selection towards optimal brain sizes. And that's also a very cool study where we've done lots of interesting phylogenetic analyses. So if the course got you excited, you can check it out. And all the data is available and the code as well, so you can go and toy around with the cerebral and cerebellar data. And I'm also super excited about our newest work, which will hopefully be out in a preprint soon. Many of my projects were only possible thanks to collaborations. So in this last part of my talk, I would like to show you some of our efforts to embrace collaboration as an essential part of our research, starting with the open sharing of data, methods, but also developing the platforms that allow us to work together. And so the data you've seen throughout the slides is openly available, for example, through our Brain Catalog project, which is a collaboration with the Natural History Museum in Paris, where we scan their collection of brains and make all the data freely available. So we already share more than 40 species online for collaboration and with a simple interface to segment the data together. But comparative neuroanatomy data is not only challenging to analyze because of the species relatedness, but also because many species, there's just no atlases available yet. And so we segment the neuroanatomical structures we are interested in by hand. And so we built the tools for scientific collaboration, like for example, BrainBox, which you can see in action here on screen, a web app for real-time collaborative brain annotation and segmentation in MRI data. It's like Google Docs, but for neuroimaging. And you can see us work together here. And we built a similar web tool called MicroDraw for collaborative annotation and segmentation of ultra-high resolution data. And it's all working on the web. You don't need to install anything or download anything. And since brains are floating in jars, can come in any possible rotations, we built Reorient, a web tool for intuitively reorienting and cropping MRI data. And another web app that we built is Threshold Man. So it's an application for segmenting MRI data just on the browser by interactively creating adaptive thresholds. And with this, I hope you now have a nice overview of brain evolution in the context of phylogenetic comparative analyses and how to do them. And with the data and tools that are available in the different resources and the R code that accompanies this course and in the Jupyter book that Austin is compiling for all of us. And on this note, I'd like to say that it's just amazing to be part of such a wonderful open science community where it's fantastic to move forward together and to draw together and segment together, to think together, to add more and more species to our data sets together and to collaborate as a community. And with this, I would like to thank my amazing collaborators who I had the joy of working with over all these projects, in particular Roberto and Nicola, who I work with very closely. And now Austin will dive a bit more into detail into some of these wonderful community efforts. Thank you very much for your attention.